I am Andrea Caporale. I briefly introduce myself. Thanks for your kind presentation introduction. I am a chemist and pharmacist. I degree in Padua and in Naples. And I am in Institute of Crystallography since 2019. But before I worked in Naples in a group of Institute of Biochemistry and Bioimaging, that is a group divided in expression of protein and the study of structural study and synthesis and peptide synthesis. Before that, I stay sometimes in German, and I have my postdoc degree in, PhD degree in Padua, and then I graduate in Padua before in chemistry. And the last foreign experience was in Dublin, where I studied the expression of protein, and I work on organic chemistry for the synthesis of special amino acids. Today, I would like to introduce an example of our approach and the tools used for that approach in the study of structure and activity of peptides. In this particular case, is a peptide that is responsible of that has an activity, antimicrobial activity. So, just in the background. In Trieste, I work together in collaboration with the University of Trieste, and I joined with the group of antimicrobial peptides. That group is an historical group in Trieste that study the antimicrobial peptides. In particular, one topic of this group is the cathelicidins. The cathelicidin was discovered in Trieste 40 years ago around, and that is a very interesting family because it's in a family of host-defence peptides, ubiquitarious in invertebrate animals. They are characterized by a well-conserved pro-region linked to a structural height variable C-terminal antimicrobial region. That means that is synthesized, that is expressed as a protein, and at the end that is cleaved, and the C-terminal that works as antimicrobial peptides is secreted. The importance of antimicrobial peptides is very important now for the problem of drug resistance against antibiotics. So that peptide could be very interesting because that is the first line to the defense of infection, but at the same time are an feature to immunomodulatory toward immunomodulatory and inflammatory activity. And that is family for the humans is expressed just one type, that is LL-37, which has orthologous in all placenta mammalians, but that kind of family is very different in the structure. In fact, for LL-37, we have an alpha helix structure. Instead, for analogs, that is RL-37, is a completely random coil. But on this point, we come back. The cathelicidin families has some characteristics and some structural feature that could be summarized in this way. The size is only around 37 residues. That is a characteristic of amphipathicity is around 65% of the structure. The charge is height variable, but the average is around 6 positive charge. And there is an equilibrium between attraction and repulsion in charged residues that is correlated with the structure that is generally they assume an helix when they are in membrane, and that helix is stabilized by the salt bridge. And generally, the charged residues is found in position E, E+3, or E+4. I would say before that human LL-37 and rhesus that is in Macaca, mulatta, cathelicidin is very homologous, and the homology in this case is at 68–70% around, but the structure obtained in different conditions, solution anionic membrane in presence of anionic membrane and neutral membrane, is completely different. You can see the spectroscopic LL-37 is completely random in salt solution instead of LL-37 is in helical. Then in anionic membrane, both are in alpha helix structure, and the neutral membrane LL-37 came back to be just a moment, come back in random coil, and for L-37 we can see that is an equilibrium between the two forms. So that characteristic could be correlated to works. Okay, could be correlated with a mechanistic action that here is reported in an atomic force microscopic analysis, and you can see that LL-37 in presence of anionic membrane created a pore. For LL-37, they create a sponge-like effect on the membrane, and that could be correlated with an aggregate form for LL-37 and in free form of LL-37, and that the interaction on the membrane is completely different. Another experiment carried by Professor Packer in a group university that is analysis of the pore formed on the membrane. She used propylene iodide and the dextran labeled with Fitch. So here there is the aspect of flow cytometry, and you can see the signal of propylene iodide and fluorescence dextran, and you can see that for LL-37 that we know that there is an aggregation, we can find that a preference of fluorescence dextran 4, that the diameter is around 3, so create a defined pore. For LL-37 that we know and we observe that is the damage of the membrane is a sponge-like damage, the only fluorescence label is the propyl iodide. Then we in our laboratory they synthesized, just a moment, they synthesized a double and a dimer, and the dimer is enough to create in a big pore, and you can see that the fluorescence Fitch dextran in a different condition, and we find is the big pore, and you can see inside the Fitch dextran 40, that is the biggest. Up. So we study the effect of salt bridge in the stabilization of structure, because we know that LL-37 is an amphipathic helix. So the first approach that is changed the charge in the sequence in order to destabilize. And what they find? They found that the structure still be alpha helix. Obviously, the decrease, and you can see the effect of the decrease of structure in this CD, but we still have an aggregate form on a structural form. And in a study of SPR, it's possible to observe that the oligomerization is reduced, but still occurs. So we come back, and that is our new approach. We came back and we started with an analysis, a statistical analysis on a library of 54 for primate cathelicidins. So we introduce a double cutoff. The first cutoff is to collect all residues present more than 80% in the library. The second cutoff is collect the residues present more than 65%. In this way, create a new sequence that we call originator. And we found that the sequence is present in 2 primates in this library, so it's native. The second point is to compare this one with the 2 sequences LL-37 and LL-37. Okay. 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 So in this way, we are able to compare the single residues, and we observe that only 6 residues is very important in this effect, in this point, on this point of view. First of all, this one, you can see that there is a RL, the first residues, position 1, R for RL-37 and L for L-37. We decided to maintain that because it's the label of the peptide. The other residues, interesting, is that the position 4, 15, 16, 29, and 36. So we decided to introduce these residues in RL-37, and we, at the beginning, concentrated in the middle of the residues. So we synthesized the RL-37 with the substitution in position 15 and in position 16. That is the approach is able to guarantee the charge, because the charge is not changed very much, and we observe that there is an effect on the structure, but we see later. In the second structure analysis, we are able to observe that RL-37, if the intensity is not so big, but there is an intensity of alpha helix structure, then we analyze the stability of this structure using the melting point, and that is reported in the table. You can see that RL-37, key 15, key 16, has a melting point around 60 degrees. That means that it is stable and that the stability is comparable to LL-37. And then we do an antimicrobial potency that was carried, was determined via MIC assay in 20% and 100% of Milton. The interesting, we decided to use just 2 strains, the standard state for Escherichia coli and standard state for Staphylococcus aureus, an example of gram-positive and gram-negative. And we found that RL-37, we just know that is a very broad spectrum instead of LL-37. LL-37, I repeat, has an antimicrobial activity, but have also an immunomodulatory activity. The substitute peptide still be broad spectrum against gram-positive and gram-negative. So the point of the analysis carried out is the helical stability. We use the melting point for that. We observe that it is important and the network of salt bridge, in particular in the N-terminal part of the peptides, in order to obtain the alpha helix structure. There is some residual, crucial residues that D4, key 15 and E16, and in particular there is probably a synergic effect between these residues. And then to study the effect on the structure, we starting to study the structure generated with AlphaFold 2. Here there is a table in which all peptides is presented in alpha helix. And at this point we can discuss, and we use the AlphaFold to identify the residues that create a network of salt bridge. And we found that for LL-37 we have a network of salt bridge and a knocked network. For LL-37 we find a very defined zone and not a knocked salt bridge network. When we introduce in position 15 and position 16 lysine and glutamic acid, we still the last one, we observe a new network and a broad network and a knocked network. That means when we can find a rationale in this point, for LL-37, we have a melting point at 68 degrees, and we have this kind of very broad network and knocked network. For LL-37, we not have any melting point because in at 20 degrees, we are a random coil. When we changed LL-37 and we remove a charge, we observe that the network is maintained, but the stability is less, and we have a lower temperature. For G16, we still be this effect, and the network is reduced. When we change it, we maintain three position, three zone where is the salt bridge, but it is not so stable, it's not knocked, and we have a low temperature. When we introduce the position D4, we not have any effect. But when we introduce the position K15 and A16, glutamic and lysine, we observe another, again, broad network of salt bridge and knocked block, and the temperature grows up. So we can do some conclusion about this work that is a new approach of this point. The first one that is an evolution between the two peptides, and that effect of evolution we are able to do when we define the originator in order to put who is before, which is before and which is after. Then we have an effect. The role of this peptide is depended by the tendency to oligomerize. Then the salt bridge is important to stabilize a form, the free form and an aggregate form. Then the engineering is not easy because there is a synergic effect between the residues, just not one residues, but a system of residues introduced can change the structure. So the oligomerization likely depends on a complex system of interaction, and then the information that we collect that is important for a new design of therapeutical peptides. And so we come back to our approach in general, what kind of tools we use. The first one is the computational analysis. Is useful to determine peptides in genome, and we use the statistical and prediction structure programs in order to correlate the data. Then that is, I try to represent a flow map rationale as our approach. Before the synthesis of peptide, cleavage, purification, biological test. When we have purified peptide, we can do the structural analysis, and we can create a biological test with the structural characterization in order to do a structural activity relationship, and then design a new peptide to come back. What we can do. First of all, the synthesis. With the synthesis, we have the facility in the university, and then we can use a microwave peptide synthesizer. That is just one line, but for our, in that point, it's okay. It's not able to do more line of the synthesis, but I repeat, for actually, that is the first point is okay. The problem is the purification, because we not have an autonomous purification. But the biological test, we are completely free from other collaboration. We are in the collaboration of the university. The group of antimicrobial peptides are able to do all preliminary biological test on escape peptide bacteria, stains in human cell line. They are able to do minimum inhibitory concentration, the minimum bacteriological concentration, potency, efficiency, and the toxicity, and all analysis using flow cytometry techniques. Then you observe the SPR, and the SPR we are able to do in our group with my colleagues, so we can integrate all the experience. About the structural characterization, we can do the CD, both in university, then in area science park. And then we think to use the collaboration with Kaminsky Institute in the future to do an NMR structure about this peptide in membrane or in other environment. So for the SARS, we can use the knowledge in the university or the knowledge of area science park, and so start to design new peptides. About the purification, that is the point that the use of PNR allow us to be autonomous, because the idea is to buy an UPLC high resolution mass spectroscopy that is useful for the analysis of peptide. In our case, it is necessary to know what kind of peptide I have, to know exactly the mass, and just to a little fragmentation in order to observe what if the sequence is correct. But this kind of instrument could be useful to the analysis of the protein, and to do some preliminary study eventually, and then move with the collaboration with the university, because with the PNR, they both in a high resolution and a very high level mass for the analysis and for the structural analysis of protein and library. And then the HPLC preparative that we asked is useful just to purification, and is not necessary to use the facility present in university. And so there is no crowd list. And the last one things, I want to thank you for attention for that one. I'm sorry for my English, probably is not so good. And thanks for the university group of antimicrobial peptides, Alessandro Tossi, that is the professor, the other professor Sabrina Paco, and the group of all persons that work on this this topic before me, and Dottoressa Paola Storici started on this topic too, and all several undergrad students. Thanks.